So unfortunately, I wasn't able to put everything up, but I will shortly after class. Just indicate what we'll be doing today. I did put up the lecture for last time. I'm actually going to perhaps replace. I have several things to post, and I probably will. I'll probably put Chapter 7 up here and just put a link here to Leisha's Creating Our Packages paper over on the left-hand side. And then today I'm going to be covering the last part of Chapter 5. And so they're potentially, I think that the next homework is actually going to be creating an OR package based on the S3 classes that you did. And so I'm going to look at Chapter 7 briefly today, but that's only a little bit of code in Chapter 7. And then I'm going to post the assignment, which will be out here someplace, which will be the fourth assignment, and that will be creating an OR package based on S3 classes. And I'll come back on Monday and talk a little bit about what I want you to do, and I may have to talk about a few topics that I didn't cover very thoroughly, and well, you probably need a review anyway, but I want to talk about the save, which lets you create a data frame, and then you save it as an object that you can actually then put in the package and have documentation about a data set type object. So I'm going to go through that with you. So on Monday I'm going to come back and do a little bit more on our packages, and in particular, but I want to do it in the context, okay, here's your homework, this is what I want to do, so that I make sure that you know the different steps that you have to do. So I'm going to review the homework, and in the process I'll go into a little more detail about some of the things you need to do. But then today we'll do Chapter 7, but we're also going to go back and pick up that last part of Chapter 5, which gets into more bioinformatics. But I can't cover the last part because I didn't realize I didn't load one of the packages. So there'll be a little bit more that I'll finish up on Chapter 5 on Monday. So on Monday we'll finish up a little bit on Chapter 5 and do a little bit on packages. At that point, we will have finished up through Chapter 7, except we skipped the one chapter dealing with C and Fortran programming. And we'll be ready to go into data technologies, which is the last chapter we're going to do. The only other chapter is a little bit on debugging, and I'm going to sort of work that in because if you're writing code, it helps you to know how to debug when you're writing code. So I'm going to do a little bit on Chapter 9 on debugging, and I may do some of that next Monday, in fact. But most of the rest of the semester is going to be spent on Chapter 8 on data technologies. And so somewhat within a bioinformatics context. So that's where we're heading. So let's start up with, let's go into our studio. And as I said, unfortunately I don't have the notes, but that was because I was actually working on the notes up until a few minutes before I left this morning. So So I'm going to go into 523, and I'm going to go into, first of all, I'm going to do a little bit on Chapter 7. And I don't have this sort of, I just have sort of the raw data that you can download yourself. I don't have it annotated yet. And I'll probably annotate this a little bit. Now, not all these pieces work. Basically, these are just some utility functions you might want to know how to do. And if you just say search, with open parentheses, it tells you the packages that you have that are installed. And the other way to find this out if you're running RStudio, so this is SQLite, and this is for this second one. No, it's actually the third one. Well, the bio installer is sort of the basic packages you have to install other packages in bioconductor. So bioconductor is a whole bunch of R packages written for doing bioinformatics. And this bioinstaller is typically the first thing you install, and that allows you to install all these other packages. This third one, well, the third one is, I'm sorry, the first one is the global environment, then the bioinstaller. The third one is SQLite. That's actually for doing SQL databases and so forth. This fifth one, by the way, is for doing this is actually data for doing microarrays on gene expressions using what's called the Affymetrix platform. And there are a couple of platforms for doing microarray data analysis. Sort of the more traditional way that was worked out was called cDNA. It was a way of getting the gene expression. And the reason you want gene expressions, and you actually may get the gene expression from 5 or 10,000 genes simultaneously. So, and if you think of genes as being the variables, and then each row, if you had a traditional data set, each gene would be a column, and you might have 10,000 columns, and each row would be an observation on, let's say, a patient, and let's say you have 100 patients. So you may have 100 rows, and that's N, and 10,000 columns, and that's P. And that's what's called the big P problem. See, mostly in statistics, what? N is much greater than P. P is the number of variables, and usually you have far more observations than you have variables. In genomics, just the opposite. Genomics and proteomics, you often have far more variables than observations. So, so in this case, we say, okay, we have 10,000 variables, or in this case, gene, and we're looking at the expression of genes. Suppose you have a simple design, and you just have patients with cancer, a certain type of cancer, and then controls, that is, patients without the cancer. And you're trying to find out which genes are associated with, which genes are expressing that are associated with the cancer. And so I'm simplifying this, but that's sort of the basic thing. So you're trying to find out which few of the genes are actually what are called markers. They're markers for that particular cancer. And a lot of times if you look at a particular cancer, it's not one gene. I mean, there are diseases that are associated with one gene, but usually a disease is associated with multiple genes. So you may say, well, okay, I'll just do a bunch of two-sample t-tests on each gene. Okay, so you're going to do 10,000 two-sample t-tests. Well, that's not exactly what you want to do, because we talk about different error rates, and you'd reject a whole bunch of them that are not important. If you did every one at 0.5, and if you assume that these were independent, which are not, but you would reject about 5% of the 10,000 by chance, even if there were no differences at all. So you'd reject a huge number of them. So you might reject several hundred just by chance. And furthermore, if you can't narrow it down to fewer than several hundred, even if there are differences, if you can't narrow it down lower than several hundred genes, it's not necessarily going to help you very much. If you were trying to find therapeutic treatments, you need to narrow it down to relatively few genes. So at any rate, this is the kind of stuff you can do, although much more sophisticated than I just described. But Affymetrix is one of the platforms that we use for microarray data analysis. And but bioconductor does more than microarrays. You can use any high-throughput experiment. It might deal with proteomics. It may deal with mass spectrometry. It may do with cell assays, and so on and so forth. So there's a lot in this package. There are, at this point, I think there are hundreds of packages in bioconductor. Bioconductors, for the most part, are written on S4 classes, and that's one of the reasons that I wanted you to learn S4. Again, I wanted to show you that if you look at packages, if you look under RStudio and you look at packages, this actually shows you the packages. And the ones that I currently have installed are the one at the check mark. So if I want to install, these are packages that are actually on our system. And as I said, when I tell Steve, hey, Steve, I need to have another package, that means it's not in this list. So there's no checkbox that I can put to actually load it for this particular program I'm running now. But there are a bunch of really important ones, like BioBase is extremely important, and I'll come back. I'll come back to a few of these as we get into that last section of Chapter 5. But there are a bunch. Now, these are our packages, not just for the bioconductor. These are all my packages, including bioconductor, that we have installed on the server. So search gives us this. Now, if I load BioBase, you know, I can load it, and then you might say, well, what does BioBase do? You know, how can I use it? Well, we are going to use it when I go back to chapter 5. But one of the ways you can get help on it, well, you can use the help command, of course. But you know, if I look at BioBase, then this, you know, we talked about a description file last time, right? That's sort of the first thing you have to do. When you write a package, you have to write a description file. Well, what is the description file for bio? That's all it is. It says the package name, BioBase. The title is BioBase, base functions for bioconductor. Who's it written by? Gentleman, person who wrote the book, and a few others. And then it says description, functions that are needed by many other packages or which replace R functions. So they may mask certain R functions. Then there are suggested packages, the tools package. The tools packages is what we use generally to build packages. TK widgets, it's called Tickle TK. This is a graphical interface that runs across all platforms. It derives from Unix. So if you think about it, like on the Mac platform, Mac has an interface, and on the Windows platform, Windows has an interface. Okay, so if I'm on the Mac or if I'm on Linux, are there any other graphical interface? Well, the traditional interface that has been used is X Windows with a capital X. And that goes back to NIT, and it's always from its beginning has been a client server. So, you know, I can be working on remote machines and have different Windows, X Windows interfaces to these machines, no matter where I am. Of course, it somewhat depends upon the bandwidth of the connection you have. But Tickle TK is another way of if you wanted to write packages in R that, for example, have a graphical interface, you can use Tickle TK. Now, there's something called R Commander. I don't particularly like it, but I know the guy who wrote it, and it's basically a graphical interface for R. I think it's clunky, but it's there. So if you want to Google R Commander, you can download the package, and it basically lets you do not all of R, but it lets you do certain, it's sort of like Jump in the sense that it lets you do pull-down menus and that kind of stuff. Then there are a number of packages in R. There's a package that does data mining that uses Tickle TK. It's called Rattle, R-A-T-T-L-E. So Rattle actually lets you do a lot of data mining operations using the graphical interface. Again, it's built on this TK. And then there are the Gulab ESETs is simply it's simply a sample data set that everyone uses. And what does it depends on? At least R of 2.1. It also depends upon the biogenerics. These are the generic functions that are specific to bioconductor. Some of the generic functions are generic functions from base R, but bioconductor may override those generic functions or extend them, make them specific to bioconductor. And the utils, there's a utils packages also. So it imports methods from methods and biogenerics, the maintainer, and the license, and so forth. And so you are going to write a file like this for your package. So you come up with this skeletal file that you get from running the skeletal function, and it gives you the skeletal for this. Then you have to fill in the title and all, your name and your email address and so forth. Notice that the email address is simply maintainer@bioconductor.org. They don't put a particular name. It's actually a team of which gentleman's on a team. Someone is always assigned as being the maintainer. But in reality, there are many, many people that work on this project. The maintainer is like the coordinator for all these other packages. So, so this is the description file. Then there's a user's guide. And in the user's guide, there are, if I look down at bio-based bioconductor, this gives you a somewhat worthless couple pages. It really doesn't tell you much. One thing I didn't realize is that it actually, by default, it requires the parallel package, which lets you do parallel computing very simply. And so within bioconductor, that's a requirement now. It automatically attaches parallel. So that means it's going to be there, or you can assume it's going to be there. And so you can design your algorithms, if you're developing a package, to automatically take advantage of parallel processing. So for example, if you've got a desktop machine that has 8 cores in it, 8 processing cores, you can spin out tasks to each of those. So they're all working in parallel. Now, you know about the apply function. Well, one of the most powerful ways of doing things for parallelization is using lapply. So you have lapply, which applies a function to a list, and each list has these components. And what happens is you can spin out each of these components to a different processor. So this function that you apply to this list, and then it has to work on every element of the list, that function may be quite complex, and each element of the list may be quite big. And so basically it spins out the first element to one processor, the second one to another processor, and so forth. So the lapply, a lot of parallel processing in R revolves around doing the lapply function. But there are many other ways of doing it. Now, I've mentioned to you before, I think, and the book mentions that generally speaking, if you're doing a simulation, you do not use for loops. You use apply functions. For loops are slow in R. Apply is much faster. And if you do lapply and parallelize it, so if you're doing a simulation and you use lapply and you parallelize it, you can make it many times as fast. Now, some of the things here that we're going to do today actually slow us up a little bit. So, oh yeah, as I said, this is not all that useful, but one of the key concepts in BioBase is called the expression set. And this is worth reading because the expression set is a data structure that's designed for genomics and proteomics. So when you have, when I simplified it, when I said that you have expression data, and that expression data is actually written as the transpose of what I just said. The way the expression data is actually written as the rows will be the genes and the columns will be like the patients. So instead of having 100 rows and 10,000 columns, you will have 10,000 rows and 100 columns. So instead of writing it as an n by p matrix, it will be written as a p by n. But what else do you have? Well, when we have an expression set, we have a lot more. We have a lot more than just the expression data. That's only one aspect of it. We have expression data, but we also have phenotype data. And the way we think of phenotype data is sort of like gender, but it could be we're sort of generalizing this. It's like the treatment regimen. It could be actually numeric covariates that are associated, with the expression data. So if we think of the expression data as being the Y matrix representing the outcomes, the phenotypes, the phenotypic data is sort of the X data that predicts the Y data. Or in other words, the treatments would be coded within the X data so that if I had a model Y tilde X, and I have a simple example of one-way analysis of variance, let's say that Y is continuous outcome, X is a categorical variable or a factor, then I'm doing a one-way analysis of variance, right? But that phenotypic data could involve categories of, it could involve not only continuous data, I'm sorry, not only factors representing different treatment outcomes. Some of them could kind of be nuisance of factors that I want to sort of adjust for, that I'm really interested in a treatment, but I want to make sure that I address for other things like covariates, but that might be continuous, and so forth. So if I have different patients and I'm looking for cancer or not cancer, I might want to adjust for where they live, where certain other demographic characteristics of those people, and so forth. So I may want to adjust for those things so that when I'm looking for the cancer aspects, I'm adjusted for all these other things that might be in the model. That's in the phenotypic data, and that's another part of the expression set, and that comes in as an n by p, whereas the expression data comes in as a p by n. But it's not just those two variables. You also have variables that explain the experimental design. We have annotation data that annotate the genes, and so forth. So the expression data set, the expression set can be fairly, well, here's the phenotypic data. And ultimately, like in this case, it's gender, type, and score. Well, score is numeric in this particular example. And we have actually metadata that you connect to the data, and that's everyone talks. I keep mentioning the NSA because what they say is they don't actually work with the data; they work with the metadata. The metadata is data about data. So, for example, if they listen to your phone conversation, they may not actually listen to the content. But what the metadata would be: who you called, and when you called, and where you called to. So, if you keep calling someone, in a certain country, and that country is someone that, if they're suspicious of terrorists in a certain country, then that's metadata that they look at. Okay? So they don't necessarily listen to what you say, but they infer what you might have said. That's a fine line, maybe. But so here, with genomic data, we have metadata that maybe describes more about the phenotypes and so forth. Is this a case control study? What type of study is it? And then we can also have, so we have what's called an annotated data frame, which is the phenodata, which is actually the phenotypic data combined with the metadata associated with the phenotypes. And so all of this gets bunched together. And in this case, we also have this particular thing right here is the human genome. And that we can pull in that type of information. We have experimental design information. All of this gets put together into what's called an expression set. So an expression set. And so it contains all that information. So when you're analyzing data in bioconductor, it uses these different sources of data. Okay. So you might want to read this paper. I probably will come back to this a little bit more because we should learn how to create these expression set data. It's so fundamental. There's even actually a more fundamental class than expression set as a function, but it's also a class. It's an S4 class. And there's actually something even a little more fundamental, I believe, if I go back here. Let's see. Maybe, oh yeah. The E set is even more fundamental because it includes the expression set, but it also includes other formats. Do you remember on some of the examples we talked about SNP data, singular nucleotide polymorphisms? Well, there's a type of data structure for SNP data, and that would be in this more general class, which is actually this is actually a virtual class. It would be in this more general class to describe how we organize SNP data. So the expression set was how we organize microarray data, but we have other data structures that tell you how you organize SNP data and other sources of data that you do in genomics, proteomics, and so forth. Okay, so this paper is an even more, and they said, well, why, if you look at bioconductor, the history of it, early on, it was mainly about gene expression data. But then all of a sudden, it said, okay, we have this flow cytometry lab, and they're creating all these cell assays and so forth. Well, we ought to be able to do all these analyses for cell assays and flow cytometry using bioconductors. So now there are these packages that do flow cytometry. There are packages that focus more on proteins instead of genes. Although the way you analyze protein expression is similar to the way you analyze gene expression, and so forth. So we have these different data structures that depend upon the different objects we're measuring. So the E set is more general. So at any rate, I won't go through that in more detail, but that is the BioBase package. I may come back to some of those. If I say this is just another way of capturing the packages without, I think it's the same list, without the prefix package. So this just gives us the actual names of the packages in a character string, and without that prefix package colon, the name of the package. And if you want to know where is the tools package, tools is a particular package, we can do system.file, and then the package equals tools gives us that information. So if I want a description for this, that is actually the description for tools. That's the description for base. Okay. Now notice the base is base R functions, and the maintainer is called the R core team. The R core team consists of about 20-some individuals scattered around the world. Development on R never stops, because if you span the globe, whether it's in Europe or Asia or Australia, someone's always working on R. Although some people work more than others. I don't know if this works. Yeah, I guess it does. This is the maintainer for the base package. In other words, it's subsetting. Now this does not work. I'll tell you what these vignettes are. Vignettes are it's just using SWeave. It's this documentation code that has LaTeX and embedded R code in it as a vignette that people can extract the code from, or they can actually read the document and learn how to use a package. So if you write a package, I'm not going to ask you to do this, so don't worry about this, but if you were developing a package, you would want to develop one or more vignettes that illustrate how to use your package. Because there's a documentation package, there's a documentation for the functions that comes out automatically as part of the code. And that documentation for your package simply has functions, arguments, very dry. It just tells you what the functions are, what the data sets are, what the arguments of the functions are. It doesn't tell you at all how to use the functions. But the vignette is something that you can step people through examples. Now usually you don't want the vignettes to be too long. But again, a vignette is simply LaTeX code with embedded R code using SWeave to create an executable document that explains your package. And so some people do it and some people don't. It's recommended, but a lot of people are lazy and don't. I should check to see if our package has vignettes. We have a package on, it's on Kth nearest neighbor. We have a random technique of doing it, similar to random forest. At any rate, this function package and this other function also, none of these package functions, and I don't know, exactly. I've got to do a search and find. There must be some sort of a function, but they've changed the name of the function. So I have to, I don't know, maybe I haven't loaded one of the packages. I'll have to check that. Okay. So if you do .library for capital L, it tells you where the library is. And if you do the .lib paths, it tells you the paths to your library. Now I should mention that your library can be, if you on your own machine, your library can be in two places. It can be at your account level. Essentially, all modern operating systems let you have multiple accounts on the same machine. And so the question is do you want to install it at the system level so that anyone with an account on your machine has access to the package? Or if you install it yourself, or if you install it on rstudio.stat, it will be in your account and only your account. That's why if I install it, it's in my account, and therefore you can't get to it. That's why I have Steve do it, because he puts it at the system level. He maintains that server. So at any rate, so if we look at BioBase, which we've already done, we should be able to get the namespace imports. In other words, it imports stuff from all of these places, all of these functions. Huge numbers. And notice a lot of them are .c functions, which are C code. So if you look at BioBase, it imports stuff from a lot of places. On the other hand, if I get the namespace uses for tools, it's a much shorter list. In fact, only graph. Okay. Well, these are just some things that you might want to know that might help you a little bit. I'm not going to have you on your homework do namespaces. You don't have to do that. At least I don't think I am. We talked about it, but in not a tremendous amount of detail. So chapter 7, you can read through it. It doesn't really have a lot. It's about creating our packages, but the real place you want to read is the paper from Frederick Leach on creating our packages. That's the real place that you want to read in order to do the assignment. Okay, any questions? Okay. So now, where am I? Let's see, time-wise. Okay. Now I want to go back to chapter 5, and I want to look, again, I haven't had a chance to upload this, but it will be. But I have to get, the last part of this is not working. We talked about character data in this. If you think back about what chapter 5 is, we were manipulating character data. And in section 3, we learned about regular expressions. So you learned about, you've probably forgotten it. It's the kind of thing that you have to use a lot or you will forget it. So manipulating character strings is very, very powerful. But using these regular expressions requires you, after you use them enough, you start learning the patterns and so forth. And it's very, very powerful, but it's very cryptic. So, but it turns out that if I'm looking at biological sequence data, there's other things that you might want to do, and so there have been a lot of special functions that have been written that specifically manipulate genomic or proteomic data. And so this section deals with that. Now, if I would execute this code, it would actually take a while because some of the operations in here are actually rather time-consuming. So I may just sort of step through this. So what are some classical string functions that you might want to do? Well, if you have strings, or you may have a vector of strings. So each element of a vector is a string. And you may want to find common suffixes, common prefixes, or just a common string across all of these substrings that are in this string vector. You might also want to find the longest repeating string in a series of strings. Now, these are all the kind of things that if you were searching for certain genes, what you have to understand is that in humans, these genes, these sequences are on chromosomes. And the amount of data on each chromosome is a lot. And we have a number of chromosomes. And so to actually find the information can be rather if you're searching if you're certain for certain types of sequences, and you might be searching, for example, matches, where you're trying to match patterns in these strings. You might be looking for partial matches, where you allow some degree of mismatch. And then problems get more difficult. Suppose you have this baseline species and you have another species and you want to compare their genomes. So you want to know how similar is our genome to another genome from another species. And so that can get extremely time-consuming. And that actually gets up, well, how do we align these genomes to know that, in other words, the genomes in the human are in these chromosomes and they differ from other species in certain ways. And so how do we match up searching for these particular patterns across different species? So we'll do some of that. So we'll start out by looking at the longest. We'll have a vector. Well, let me bring the vector up and maybe it will make sense. So I think this is already loaded, but I'll make sure it's loaded. So here's biobase. A lot of these string functions are in biobase. So let's look at a string. This is not a genomic string. So we have a string, and we have a vector of strings. So the first one is not now, not as hard as wow, and not something new. And so if I ask what is the longest common, can anyone tell me the longest common, and let me do this a little bit so I bring it up on the screen. What is the longest common prefix? Prefix means you begin. For each of those, what is the longest common? Not space. Space counts as part of the string. So there's a function called LC. That means the longest common prefix. And if I do the longest common prefix, it's not space. So what is the longest common suffix on the end? Well, if you look at the first two, you might think it's OW, right? But that soon gets dashed when you look at the third string. In fact, the longest suffix is W. They all end in a W. So prefix is the beginning, the suffix is the end. So if we look at this, we can see that it is W. Okay. Now, suppose we want the longest repeated sequence or substring within one or more. In other words, it might be in a single string or it might be in multiple strings. There's something called get longest substring. And that will be sort of a repeating pattern. This comes in a library called libs tree or libs tree. This is actually not in bioconductor. This package comes from Omega Hat, which is another repository of a lot of packages, particularly on the interface of computer science and statistics. So if you look at XML, which is extensible markup language, which is generalizing HTML, which is the web language. If you look at Google Maps, for example, and you look at the underlying web technology, it has all those interfaces, so you can program Google Maps. It has interfaces to all types of databases. It lets you embed, there are packages that let you embed R into this Linux database. So you can actually have a spreadsheet, but, well, okay. You can embed R in a spreadsheet, but you can also embed R in a database. So, you know, if you have a database and you're manipulating a database, usually the database companies have some fairly, you know, they've got various functions that let you operate on the database. But they're not like R functions. In fact, you can embed R in a database, and you can do very powerful operations in a database. Now, when you have big data, that becomes important because if you have big data, you may want to do your calculations in the database. It's called in-database computing. And at least with these open source data, I don't think you can do it in Oracle because it's proprietary, but with these open source databases, you can embed R within the database. And I think PostgreSQL is supported, which is a very powerful database that's open source. Anyway, so let's load this library. And then I'm going to create a string called biology, and then I want to know the longest substring. And are there any sequences in there that sort of repeat? Well, there's actually only one, and that is O, the letter O. It appears twice. There's no two sequence. There's no two-letter sequence that repeats. How am I doing time-wise?
If there were, would it list both of them, or would it just list the one that appears the most?
I think it's the longest, I think. The longest one. Yeah. So I guess, let's see. 145, okay. So we keep track. So I have a tendency to keep you too long. That doesn't mean I won't, it just means I'm checking. Okay, so the genome of every organism is encoded on different chromosomes that consist of either DNA or RNA. And so at any rate, we're going to look at a case where from DNA you can transcribe to RNA. And so we're going to look at a process that takes an RNA string, an RNA string, and you drive back the DNA string that generated it. So you can go from DNA to RNA, but we're going to go backwards. So let's create an RNA string. So that's the string. So if I do, if I do ST1, okay, so that's the particular string. This function gives you this metadata associated with it. It's like attributes. And then I have a function called cDNA, and that takes you back to the DNA from the RNA. By the way, if you really don't understand these biological terms, the book doesn't help you very much. You probably noticed that. And if you haven't had biology, I would suggest, and I'm actually going to say this, but I would suggest that you maybe Google it and probably go to Wikipedia. Because for things like genomics and proteomics, it's pretty complete. What's going to happen is you're going to look up DNA, and it will take you a place, and that's going to lead you to, you're going to have this sort of, you'll read a word in there, you'll start reading about DNA, you won't understand, and you'll link on it, and you go to another place, you read it. Pretty soon you're pretty far away from DNA. Of course, you can go back, but I would recommend that if you don't understand the DNA, which is sort of the double helix, if you don't understand that and how it works, either functionally or structurally, there's a structure to it. There are only 4 letters of the alphabet. And so we have a simple alphabet for DNA. And RNA has a simpler alphabet. I mean, it has a similar alphabet, except for one nucleotide. And so there's, you can read about that in Wikipedia, or if you don't have a particular book that talks about genomics. Now, when I'm looking at proteins, I have a more complicated alphabet. My alphabet is 20 amino acids. So at any rate, so let's convert this back, and let's look at CD. And so this is actually, notice the U's in here. This is the RNA, and this is the corresponding DNA that generated that RNA. So, so there are nice little utility functions like that. Now, this match probes, in Affymetrix, you have all these probes, and you get polymers from that. Actually, they're of length 25. And so when you're if you're trying to match these Affymetrix probes, there's a function called match probes. The only problem is I can't find this package. So this code does not work. I'm still looking for it. I haven't found it in bioconductor, so it may exist out there, or maybe they've deprecated it and they've taken this functionality and put it in another package, dealing with alphametrics microarrays. But when I talk about gene expression, alphametrics was one of the key ones that does these gene expressions. And there's a bunch of packages that relate to alphametrics, microarray data analysis. And this probes, I don't know, as I said, but haven't been able to find it yet. So these next few things, including this, relate to the alphametric stuff. Okay. So a lot of times you'd like to know the complete. If you look at the genomes that have been sequenced, there are quite a few. Now, built within R, there are a number of species that have the whole genome of those species. So you can get the whole genome in R and manipulate them and compare the genomes of different species. Okay, so, but R doesn't have all of the genomes that have been sequenced. I don't know if it has 30 or 40, but it does, I think we'll see a list later on. Of course, it has humans, but it has a lot of other species besides humans. So basically, complete genomes are represented as DNA string objects, and these rely on certain infrastructure in what's called the BS genome package. But that package is sort of that package is a base. It's a package from which all the other genomes have a sort of a sub-package. So in other words, there's a human, the BS genome is a package, but there's a BS genome package specifically for humans. And there's a BS genome for other species too. But they all begin with BS genome dot something. So at any rate, so here's the human one, library. Notice it's BS genome dot H sapiens. What do you think that H sapien? You know what it is.
Homo sapiens.
Homo sapiens. That's us. Dot, and then this gets into, I think the UCSC is probably the source of this, and then the HG18, HG for human genome, version 18, and it's, I don't know if the latest version is 18 or 19, but in other words, every few years the human genome gets updated. You might say, well, isn't it static? Well, you see, not exactly, because if the human genome were exactly the same and static, then we would look all the same. But we don't look the same. So there are differences in these nucleotides and so forth. So there are various differences within the genome. And in fact, there's an X and a Y chromosome, right? Does anyone know what that is?
Male and female.
It's the male and female.
So who do they pick? Whose genome?
I actually have this. Well, I think that they actually have both. They have in fact, they do, because I actually. So I'm going to load this human genome live. This loads the whole genome for humans. And so if I print H sapiens, then this is the structure it has. So what do you think CHR1 is? Chromosome 1, chromosome 2. Here's the XY chromosomes, the sex chromosomes. Okay? And then these are sort of some subsets that have, I don't know exactly what all these random sequences deal with. But so if you might be studying chromosome 22, so you might be focused on a particular chromosome. And the amount of data on each chromosome is a lot. These chromosomes have a lot of stuff packed into them. I mean, we're not that simple. So, you know, we have a lot of information stored within each chromosome. And there are regions of this chromosome that we call genes. And, but then there's regions between these genes and so forth, which, you know, there's a lot of research going on, not only about the genes and diseases tied to certain genes, but also sort of what's all this stuff between the genes.
It's like most of it.
They used to call it junk, but it's funny now. It's not exactly junk. There's a lot of good stuff in there. Yeah,
That's like most of it, right? Most of the information in the genome is like non-encoding.
it's non-encoding. When we say encoding, when we're talking about the parts of the chromosome that are genes that are encoding, that means encoding for proteins, because that's what creates the proteins. And they do all the work in the cells of the body. Without proteins, you have history. The proteins are what do all the work in the cells and so forth. So you have this process, of going from, gene, which ultimately ends up manufacturing proteins. And how do you regulate? There are certain genes that regulate. There are certain regulator genes which regulate how much proteins get generated. So if you suddenly run, or you get afraid and you run because you're afraid, certain things are stimulating these gene regulators that say, hey, I need more stuff here. Or if you're doing distance running, I need a little more help here, and so forth. So there are a lot of regulator genes, and so it's a very I'm not a biologist, but this is a very interesting area. If I look at the available genomes, and that's what I was talking about before, each one of those is we're in there somewhere. Where are we? H. We actually have the human genome HD17 and HD19. So the HD18 is actually an old one. I think the most recent is 18. I presume that's the most recent. But for some reason, it looks like they've deprecated. No, here's 18. So they have 17, 18, and 19. Those are different. 18 is a more recent version of the human genome than 18 and 17. But if you were looking at sort of the historical development, obviously there was also an HD1. Back, when was it? In late 90s. And when they said that we've sequenced the human genome, they really hadn't completely sequenced it. It was substantially, but they didn't know a lot about mutations, and there was a lot of information that was still missing. They didn't have all this information about SNPs, and so there was a lot missing in those early days. And so the picture of the human genome is growing it's growing by leaps and bounds every year. So there's this constant update of information, and not just the information in the genome itself, but all the annotations, all the metadata about the genomes. So ultimately, you can think of this gene interaction network. So if you think of a graph, and by a graph I don't mean what you're thinking, I mean dots and edges. So if I have 2 points and an edge connecting them, it means there's a relationship between those two genes. So if you think about Facebook, and each of you that are on Facebook, and I'm not in that picture, there's a dot for each of you, and then your friends have a line between you and them. And so you have these networks and sub-networks within these large networks. And so the question might be as well, how do we analyze network data? Well, here we have gene networks because certain genes co-regulate other genes. And so we have these linkages between genes where certain genes act in parallel. And that's the thing about cancer. You may have certain genes that are acting in parallel that generate, that cause the underlying cause perhaps of the cancer. And so, so we have these huge gene interaction networks, but we also have protein interaction networks. And so these are huge, because we have 20-some thousand genes? It seems to not be a known number exactly. But we have, we have maybe 20-some thousand genes. There are actually some species that actually have more genes than us, but our genes are more complex. At any rate, so let's look at chromosome 22. The problem is you may want to mask out information from a particular. If I'm looking at chromosome 22, there's a lot of information I may not be interested in, and there might be a lot of slots that have this N. And the N is basically certain spots where you basically don't have information about. And so we can mask it out by using a mask function, and that actually makes this sequence well, it masks out those particular ones that have a particular. Notice that in right here in quotes, it says, this is H sapiens $sign, right? We're extracting. And we're extracting the information from chromosome 22, and we're going to mask all the ends, which will make it shorter. So we'll do that, and then we'll look at it. Oh, I shouldn't have done that. Now, this is the nice thing about bioconductor. It doesn't print out the whole. You can get it to print out the whole thing, but it uses these dots to say, okay, this is part of it. So this says a view on a 49,691,432 letter DNA string. That's once I mask out a lot of the sort of non-important information. Once I mask it out, and I'm looking at only one chromosome, I still have over the string length, the letters is over 49 million long. So now you can begin to get the picture about why they call genomics big data. It's big data for a couple reasons. Well, if I've got 23,000 genes and I'm doing gene expression and I've got a matrix that's 100 by 23,000, and I am trying to relate the genes to the phenotypic data, which is what you're trying to do. You're trying to figure out, you look at the phenotypic data, and that will include the treatment information, the experimental design, and it may be a more complex experimental design. So I have this experimental design, and that's part of the phenotypic data. Then I may have covariates and all this stuff. And I'm trying now to do an experiment on 10 or 20,000 genes to figure out which ones differ according to this experimental design. So that's a little different than your Stat 512 course where you have one Y. You may have a complex design, but you still only have one Y, typically in Stat 512 at least. If you get to Stat 545, you got multiple Ys. Stat 541, the multivariate. So if I talk about high-dimensional data analysis, well, this is the kind of stuff I'm talking about. At any rate, so this is the chromosome 22. This is a partial list. This gives starts and end sequences. This is now separated because of these masks. And this tells the width of each of these pieces. Now, some of those segments are quite long. Some of them are in the millions. This I don't think works, and this frequency doesn't work. And what they've done is the alphabet frequency actually, if I run this, it doesn't work. But if you actually look in packages, and I think it's in Biobase, and now down here I can look at all the functions. So I had a function, I thought it was in here. Okay, so if you don't know where it is, apparently it's in another class. Does anyone see alphabet? No. So if I have ALPHA, BET, FREQE, frequency. So if I'm looking at the frequency, oh, it's in biostrings, not biobase. So if I go back to packages and I look at biostrings, then I can go down here and I can look at, and I didn't have time to sort of figure this out, but obviously I've looked it up before. So there's the function. Now when I looked it up, it actually said letter frequency. So the question might be is if I change it to letter frequency, it might work. But the problem is I didn't see, if I look at the arguments, I didn't see an argument down here that said FREQ. So the problem is if someone, I have no doubt that there's a function in bioconductor that does this, but the problem is that that function no longer, it may have been switched over to here, but then the arguments are different. So we need to, at some point, this fell by the wayside, or it's in some other package or something. But at any rate, it takes sometimes a little detective work. That's why I wish it would update this book. It might work, but you're not going to get frequency counts, and I'm not sure the end result of this. Sometimes you do this and you get a huge amount of output. Let's see if it works. Oh, yeah, it works. In and out. That's not, well, I'm not sure what that tells us. That says you get an N with frequency 0. I mean, later on we're going to see a table. I don't know if we'll get to it today, but we'll see a table of frequencies where we're hunting for certain sequences, and we want to know how many times it repeats. And it will give a table, it will give a frequency table. It's similar to this. I don't know. If anyone plays around with it and discovers how to get it to work, let me know. Okay. Well, now, matching patterns is one of the key things that you want to be doing. And there are a couple functions that you might want to use. You might want to exactly match a single query sequence against a single reference. So a lot of times you may have a reference species or a reference sequence of patterns, and you have a query sequence, and you want to know how that matches within this long sequence, perhaps. And so there's a function called match pattern. And then there are some of them that match a pattern that have that allow a gap in between. In other words, you match on the left, and then there's a gap, and you match on the right, and the gap you specify up to this distance. And so, so that allows sort of a match here, and then it's almost as if you were hunting for a gene here, and you have stuff between it and another gene. That's typically not what you might be doing, but it's sort of the idea. But typically you have, that's the match RL means right-left pattern. And then suppose you want to compare, suppose you have a huge number of sequences, maybe from a lot of different species, and you want to compare them all to a reference species or a reference sequence. And that's called a match PDEC. And so those are three of the functions. Now, just as a background, now a motif is a short sequence that occurs repeatedly in a group of related DNA or RNA, or it could be repeatedly appearing in a protein or peptide sequence. So let's suppose we have a simple sequence data, TATA. So that's our sequence, and we want to match. We want to find out how many times DATA appears in chromosome 2 without the ends. Okay? So I'm looking at chromosome 2, and I want to know how many times the TATA. You see you have a TA, and then you have a C and a G. Those are the 4 letters. So T and A are 2 of the 4 letters. So TATA, that's a sequence. How many times it appear in the chromosome 22 of a human? So let's now oh, that was faster than I thought. So let's take a look at it. So we can look at it. Now, fortunately, it has the ... because I found it, I found it starting here and stopping here, and I found it in 5276 instances. Okay, and actually, here's TATA, and it keeps actually going. It's finding repeats of the A. But that's from that 49 million and so that it searched it, and it found 5,272 instances of it. Now, you might ask yourself, how are we doing time-wise? Okay. You might ask yourself, well, how many times do particular patterns appear? So if I run this, well, I'm sorry, this is the 72. That's just this last one. Okay. I'm getting ahead of myself. Okay. Now, suppose you say, well, I'd like to have mismatches. I'd like to allow 1 or 2 mismatches. I don't necessarily. I know there can be mutations where you can have insertions or deletions of nucleotides over time. If you're looking at it evolutionarily, over time you can have insertions or deletions of nucleotides, or you can have mutations. So you may not get an exact match because of SNPs and so forth. So I need to be able. I want to find a partial match. I want to find a match that maybe has a few mismatches in it. So we're going to look at mismatches. Now what we're going to do is we're going to look at TATA in the same genome, but we're going to allow 1 mismatch. Now, are we going to get more or less? You're going to get more. Because if you say mismatch equals 0, that's just what we did. So we're going to find potentially a lot more sequences that allow 1 mismatch.
It could be anything, right? It could be a G, C.
It could be anything. I'm not saying what this mismatch is. It could be any one of the alphabet letters. Okay, so let's look at this, and let's see how long it is. Well, 102,000. We went from 5276 up to 102,104. I mean, I went by a factor of 20 by allowing 1 mismatch. Now, granted, my sequence is very short, and so I'm with 1 mismatch, you know, with a short sequence, it's going to allow a lot of other possibilities. So at any rate, I'm not sure what this is. Not sure what this is giving. This is giving a list.
The first 3 mismatches.
Maybe. I need to read about it. I mean, these are components of, it's giving a list, and the first, second, and third components are 2, 5, and I'm forgetting what those are. Oh, yeah, we're only giving the first 3. Yeah, because, yeah, you wouldn't want, yeah, you would not want, I think it's maybe giving the starting position. You would not want to do all 102,000. It would be scrolling through your console for a while. That's only the first 3. 1:3. Okay. Now, suppose you want to match a huge number of query sequences to a single target sequence. And this is the type of stuff you may do in a lot of the using high throughput experiments. There's something called genome-wide association studies. Then, you know, you can create huge amounts of data very, very quickly. So we're going to look at some Affymetrix data. And I'm going to look at the sequences here. Now, it's always a good idea to maybe, before you print something out, it's maybe a good idea to look at how long it is. So this is some Affymetrix probe data for doing microarrays. And it turns out this has over 200,000. So I'm going to compare 200,000 sequences. Now these sequences are 25 polymers. They're called 25. If you think of polymers, P-O-L-Y-M-E-R-S, we call them 25-MERS, meaning it's a polymer of length 25. And the Affymetrix use polymers of length 25. So their probes are giving you sort of these lengths of 25 each. And I want to know, so I have all of these sequences from this probe data, and I want to compare it to a reference data set. And in particular, I want to compare it, well, let's see how many unique characters I have. Okay, I have 25 unique characters. I'm going to look at the first, this is the first 5 of each of these is 25 long, and I'm going to look at the first 5 of those. And then I'm going to compare it, okay, not yet. I'm going to do the p-dictionary, and that's going to form an internal data structure, and I think this tells you that this is a type of object, and it's, again, the 2001-800, and it's a width 25, because each of these is going to be 25 long. And then I'm going to use match p-dictionary. I'm going to match p-dictionary to chromosome 22. So I've got 200,000, over 200,000 sequences, and I'm going to compare each of those to chromosome 22 to find the matches. So I'm taking these probes from these Affymetrix experiments, each of which is a polymer of length 25, and I'm going to compare each of those. So I'm comparing 200,000. Now, I forget, this may take a little while. Okay, so I'm doing match P dictionary, and so we're waiting. I can't remember. Yeah, that wasn't too long. So let's see the length. That's sort of the length, but let's look at what we want to know is we're going to get a count index, and I'm not so interested. I'm interested in a table. The table says that of these 201,000 sequences, 198 found no matches. There were 2,855 that had one match, and so forth. And there's one sequence that had 2,771 matches. In other words, there was one probe that had a huge number of matches on this chromosome. And then there's another probe that had 2757 and so forth. So this is just a summary of how many times each of these how many times we found a match for each of these probes. So that's kind of, it says one of the 25 MERS matches 271 times for this chromosome 22. And I'm almost finished here. This just confirms that is the string that matched. In other words, here I'm looking at the max count. I'm looking at the max. This is the string that matched 2,771. That is the probe. And then if I count the pattern, that's the 2771. So this is confirming what is in that table. So one of those probes appeared many, many times on chromosome 22. Okay. So I'll stop here. Does anyone know what a palindrome is?
Same forward and backward.
Yeah, it's a word that's the same forward and backward. So madam, M-A-D-A-M, spelled the same way forward and backwards. Turns out, but this is really important in genomics, but we kind of generalized the notion to say, okay, we have this first part, we have this middle part, and we have this last part. And it turns out that if the first part matches the reverse complement of the last part, that is a secret. In that particular instance, we can have hybridizations. And so there are sort of biological things that you might search for if you're searching for hybrids, like when you have different corn variety hybrids, for example. And so, at any rate, we'll stop here. And this section I can do. It's the next section I have. The next section is the final section on alignment, sequence alignments, which are very powerful features of. If you want to compare two species and how they align their genomes, then sequence alignment is very important. And you can have both sequence alignments where one species with another, so you can have multiple alignments if you're comparing multiple species. And so, at any rate, that is the next section, but I don't have the package yet, but I'll get it soon. And when I do, I'll be posting this on the website in the next few days. Yeah.
So any chance you could send out a list of those packages?
Yeah, that was part of the problem. If anyone can tell me if there's a place in the book that has the list, what I'm doing is I encounter them, and so they're kind of spread out through the book. Now, maybe in the preface or somewhere, he does list them all, but I didn't see it, but maybe it's there. But if you do look here, the problem is some of them aren't in the book. Only some of those came from the book. You can kind of tell which ones. But if you go into our studio and simply look at this, you can kind of get an idea of what you need. if you're doing it on your own machine. Okay. See you Monday. I'll be posting the next assignment, and I've told you what I'm going to do next Monday.
Hey, Dr. Harner, before you close out of that, if you scroll up, I have a quick question. Where it's like it's about all the extra A's at the end of the sequence.
Oh, this one.
Yeah, you're looking for Tata.
Yeah, you actually are looking for 7, yeah.
So
Yeah, you are looking for the sequence. They call it Tata, but they're actually looking for TATA, AA, AAA.
okay, all right.
You actually are searching for that.
You're searching for those extra A's.
Yeah, I had missed that. Yeah, Tata is the name, but the actual sequence has those extra A's.
Is the final?
I need to look it up and put it on the. If it's not on the syllabus, I'll look it up and post it.
So,
I need to.